All right, so now that we've learned a little bit more about laboratory biotechnology, let's focus a little bit more on things that we may see out in the field, reproductive biotechnology. So before we get in too far, I want to emphasize the importance of knowing some specific key vocabulary terms related to cloning, okay? First is obviously clones, individuals with identical genetic makeup. So Dolly the cloned sheep was our first mammal to be cloned, and that was in 1997. And cloning always begins with just a single cell. We'll talk a little bit about cloning here in a few minutes. Surrogate mothers are cells that are harvested and grown inside of the embryos of a female on behalf of another female that may have reproductive complications. And then, of course, we have our genotype and our phenotype. Genotype is the genetic potential are passed on to an offspring, and phenotype is the final visible characteristics that we see. So a key equation that I want you to know from this is phenotype equals genotype plus environment. So our phenotype is highly influenced by the environment in which an animal is surrounded by. So for example, let's say that a specific gene in an animal is feed conversion that's passed down, and we're trying to breed for effective feed converters or feed efficient animals. Okay? If we don't feed our animals anything, that's an environmental issue, and we're going to have animals that are not effectively utilizing their feed conversion traits. So we want to make sure that we are providing a healthy, good environment for animals so that they can have the best possible phenotype possible. So some key reasons for cloning animals. There are a few here that I want to highlight. First, genetic superiority. When we clone, it helps us maintain positive genetics in a faster manner. Second, animal and product uniformity. Chicken wings is a prime example of this. This helps reduce genetic diversity so animals can receive the same treatments and helps keep our food animal products uniform and appealing to consumers. Third is endangered species, protects populations that may go extinct. Fourth is sterility. This allows us to maintain the genetics of animals that would otherwise be sterile. So for example, mules are naturally sterile. So cloning helps ensure that we help continue to have mules. And lastly is research. This establishes control groups for different experiments that we may have relating to animal biotechnology. Our next slides are going to talk a little bit more about the specific steps of cloning and how cloning processes work.